Somenath Rai to the audience. He is a principal scientist at CSIR-CGCRI. He received his PhD in materials science from Indian Institute of Technology, Kharagpur in 2004, and subsequently joined Swedish Census Center, Linköping, Sweden, as postdoctoral research fellow. He worked there for 2005 to 2007, and he has worked as a coordinator research programs at Florida International University, Miami, USA. He joined Institute of Bioengineering and Nanotechnology, Singapore in 2008. Worked until May 2012 as a research scientist in biosensors and bio devices group. And he has joined CSIR-CGCRI in June 2012. Dr. Roy is a recipient of National Scholarship of Government of India. He was featured in Nanotechnology Thought Leaders Series 2010. He served as a member of Review Committee for National Science Foundation NSF USA and visited University of Central Lancashire, Preston, US as a distinguished collaborator in 2019. Dr. Roy has co-authored 2 book chapters, and he has to his credit for 40 research papers in peer-reviewed international journals. And he has to his credit granted 3 United States patents, 4 PCT, and 2 Indian patents. Dr. Roy's present research interest is focused on synthesis of nanostructured materials, surface functionalization and fabrication of micro and nanoscale chemical sensors and biosensors. His landmark research contributions include first ever experimental demonstration of charge transport through a single DNA molecule using a uniquely designed carbon nanotube based platform, and design and fabrication of mass producible nano gap sensor array using standard microfabrication technology for ultra sensitive sub-femtosecond molar detection of DNA and mRNA, and development of microfluidic enabled microarray for a detection of miRNA with a single molecule resolution under optical microscope. So with this, I invite Dr. Somenath Rai to deliver his today's talk. Dr. Rai.
Thank you very much, Dr. Annapurna, for your very kind introduction, and I'm also thankful to Dr. Juhi Chakraborty for inviting me to give a seminar talk in a webinar, especially which is focused on biomaterials. All right, so I work in a slightly different field, which Dr. Annapurna has already detailed. So I work at the cutting edge of molecular biology and engineering. My today's talk will focus on molecular bridging of nanogap. Since the slot is 15 minutes, so I have tried to focus on a particular topic, and this particular device or the platform is envisioned as a futuristic approach toward PCR-free rapid detection of nucleic acid biomarkers. So with the prevailing situation of COVID, our society has very much understood, which were kind of ignorant apart from the experts about all those PCR technologies and so on. Now in the newspaper and everywhere, we're talking about PCR. But the limitation of this PCR technology sometimes is the turnaround time, because the sample needs to be transported to the laboratory. And we have seen that in practical scenario, in a pandemic situation, the turnaround time could be up to 72 hours and things deteriorate. So the world is now looking for reliable point of care systems where the detection can be done at a clinic or in a resource starving environment, or maybe at home, most preferably, and we can transmit the signal through our mobile phone or other like internet devices. So since there are several technologies available, like optical, electrochemical, or other molecular biology-based techniques. Still, our focus would be on electrical sensors. So this is where this, this sounds a bit, too focused, like molecular bridging of nanogap. This looks like a very specific topic. But the thing is that when we talk about electrical signals, then we have to make certain devices, which is electronic device, and we have to bridge or we have to detect the biomolecules which can generate some electrical signal upon biomolecular transduction. So with this,
yes, your slides were not moving. Yeah.
Yeah. So now I get into the genesis of this problem. Why did we see? Now we are talking about POC, but when I started this research, it was from the biophysics point of view. Why did we start this research? If you look at the publications in early 2000s in all the big sort journals like Science, Nature, Physical Review. So I was reviewing through this journal quite some time ago when I was in Motorola's nanofabrication facility. So we are looking at the DNA's electrical charge transport, and you see, I mean, here I have given the snapshots of only 4 selected papers, and you see the variety. In some studies, it is found that DNA is an insulating material. In another study in Nature, the curve we can see is talking about a conductor, like a metal-like conductor. In another study in Nature, I think it's pretty close, within a year or so, they talk about the semiconducting property of DNA. And in another paper in Science, in the following year, DNA is termed as a superconductor. So starting from insulator to superconductor, every electrical properties have been attributed to DNA. Now, why do we want to know the conductivity of DNA? Okay, so from a biophysics point of view, yes, we want to understand the charge transport through DNA, and probably we can talk about making DNA based field effect transistors and so on. So that was the primary focus, but it has got a medical implication as well. See, DNA charge transport has a very strong correlation with oxidative damage, and this oxidative damage is the underlying mechanism of several diseases. If we talk about cardiovascular diseases, we know that atrial fibrillation or ischemic heart disease, they have very strong correlation with oxidative DNA damage, and oxidative DNA damage has strongly correlated with the charge transport. So it is not only from biophysics point of view, from bioelectronics point of view, rather from medical point of view, understanding the charge transport mechanism of DNA is very important. Now, if you find this spectrum of charge transport in Science and Nature journals like insulator, conductor, semiconductor, superconductor, so what would be the conclusion? Then we tried to review this paper in detail, and we have come up with this summary. So we found that there are several factors that influence the charge transport measurements. So it is not only the conclusion, we had to get into the detail of those measurement techniques, the substrate that has been used. I mean, this substrate is in semiconductor context, the device that has been used, and also the buffer and all this thing. So we have found that the factors, the variance among the whole spectrum, among all those papers, is that first, the DNA length. See, DNA is not a naturally conducting molecule, but in its purest double helix form, there is stacking of the bases, nucleotide bases. There's a very specific pi stacking. And because of that, there is a hopping mechanism of the charges. So when it is in a perfect double helix condition, it will have a hopping of charge transport. Now, this charge transport is attenuated if the DNA length is very long, if your electrode spacings are too much, or you have some mutation in the DNA, then of course this charge transport will be attenuated. Like the stepping stones, if you remove one of the stepping stones, of course your journey will be hampered. So anyway, so first is the length, and second is base sequences. Base sequences has got a very big role to play here. So see, base sequences, the base sequences that we know that DNA comprises 4 bases. adenine, thymine, guanine, and cytosine. Now the sequence of these bases in natural DNA, of course, that is what we call as gene sequencing. Now the fragment that we are taking for our experiment, that is very, very important. Like we know that if the DNA is enriched in guanine and cytosine bases, we know G and C makes 3 hydrogen bonds, and adenine and thymine makes, this is our school biology, adenine and thymine makes 2 hydrogen bond. So because of the low ionization potential pertinent to the guanine basis, it has been observed that if the fragment of the DNA that we are studying, if that is rich in the GC content, then of course the conductivity will be higher. Sorry. So the third point is the counter ion. See, DNA we suspend in buffer. So there are different kinds of buffer. The molarity of the buffer is very, very important because the DNA backbone is sugar phosphate. It has got negative charges, and the positive charges, like we have sodium, potassium, all those positive charges, positive ions in the buffer, and those positive ions are attracted by Coulombic forces to the DNA backbone. And sometimes these ions, they contribute, and that can be misled as the native conductivity of DNA. So we have to understand also what ionic concentration is taken for the sample that we are dispensing on the device. The fourth aspect, that is the engineering aspect, DNA electrode. Of course, this is physics and engineering aspect, DNA electrode interface. We know that when we interface a biological molecule with some electrical or optical or whatever device, engineering device, we have to be very careful about the interfaces. In case of electrical devices, at the junction point, some resistance is formed, and there is a physics phenomena called Coulomb blockade. Whoever my colleagues here who are from the physics background, they understand what is Coulomb blockade. Coulomb blockade is the effect when the conductivity is reduced with decreasing bias. Ideally, if it is a ohmic type of junction, then we know that the conductivity is constant for a given metal or an interface, but there are certain situations where the conductivity is decreased with the bias. So that is called Coulomb blockade effect. So if we have an interface which is just physically absorbing the biomolecule on the electrode surface, of course, we may observe Coulomb blockade effect, and that would severely perturb the charge transport mechanism. So some of these papers, which are more on the insulating side, they have included this Coulomb blockade effect unknowingly, and they have not realized by doing so. Okay, and the last point we have noticed is the substrate effect. As I have told, that the charge transport in a DNA molecule is facile only when DNA is in its native double helix form, and there is a well-organized pi stacking between the nucleotide bases. The moment you drop a biological molecule on the substrate, there will be interaction between the substrate and the biological molecule, and there is a possibility of substrate-induced conformational deformation. This substrate-induced conformational deformation definitely perturb the charge transport through the molecule. So these are the parameters that have been found divergent in all those papers, and hence the conclusion were very different. Okay, so this one I just briefly try to bring an analogy. The most important from engineering point of view, the most important thing is that we have to identify right kind of platform, right kind of electrodes to probe into a molecule which is having nanometer dimension from various aspects. We see in a double helix form, the diameter of DNA is 2 nanometer. The base pair separation is 0.34 nanometer, and its major and minor groups are also on the nanometric scale. And if we see this analogy, so this is the analogy I would like to bring to make the things very simple. If we want to grab a nut, we need a plier, but if we want to manipulate eyelashes, we need a tweezer. So depending on what we want to manipulate, we have to find the right tool. So for a material, for a biological molecule which has nano dimension, from various point of view, we need to have nanomaterials to probe into it. So we have seen several, again, several landmark papers in Nature proceedings of this PNS and so on. So we saw that the DNA wraps around a single carbon nanotube, or it is immobilized on gold nanoparticles, or even nanopores are used to manipulate the DNA. So most important thing is that we need to have a very fine tool customized for detection of this nanomolecule. So now I am coming to the engineering part of it. So nano architectures. So since this is a very focused talk, I don't have the opportunity to discuss in further detail. So I would say that if we want to design an electrical platform that is capable of probing DNA molecule, as I have discussed, I have explained in my previous slide. So there could be several architecture, and we have to create a nano gap. That was the title. That was in the title of my talk. Ideally, we should have a nano gap. A nano gap means the separation between two electrodes with nanometer dimension, like less than 100 nanometer. So what we can have nanowires or nanotube with a nanometer gap, or we can have planar gold electrodes with nanometer gap, or this is another unique structure that we have patented. I will show you in my subsequent slide. This is the vertical structure. That means two electrodes are separated by a nanometer thick nanometer thick insulator. So this structure we can fabricate using focused ion beam etching. So this the first one we term as top-down approach, and the later two which is fabricated either using electron beam lithography or using standard photolithography. So this is the chronology from left to right that I have done to probe the DNA molecule. So first one is top down, and the later two are bottom up approaches. Okay, so now coming to our the first architecture that we have fabricated. In this case, the first challenge was that how can we ensure that a single DNA molecule is anchored? So this was the first challenge.
You have a couple of minutes left.
Okay. In this case, we have used a pair of carbon nanoelectrodes, which basically ensured that only one DNA molecule is anchored between the electrodes, and then we have applied the dielectrophoretic forces to bring the molecules, to overcome the Brownian motion and bring the molecules and anchor them between the electrodes. And then the interfacial effect, to avoid the Coulomb blockage, we had a covalent binding between the molecule and the carbon nanotube. And of course, substrate effect. I wish I had more time to explain this phenomenon. These are very, very interesting. I told that the substrate induced deformation causes perturbation in the charge transport. So our this particular architecture, you can see that during the electrode making process, we make a trench, thereby hanging the DNA molecule between the electrodes, and thereby we overcome the substrate effect as well. So this is the data, and since I have to rush a bit fast, I quickly show that this current voltage sweep, we can see that in absence of any DNA molecule, we have a kind of almost negligible capacitive kind of effect. And when we have a single, the red one is for the single stranded DNA. And when you have the target DNA, then we have a change, discerning change in the current, the discerning increase in the current because of the formation of the double helix. Okay, and by using this method, we could discriminate between the two different kind of model genes. In this case, one from the viral gene influenza A, another from bacterial gene Pseudomonas. Why is it so? The length of the DNA was same in both the cases, but as we discussed in the beginning, that if the GC content is higher, it is sequence dependent. If the sequence is different, we would be able to see different kind of different levels of output signal. And by designing so, by implementing so, we can distinguish between the like different kinds of genes from different pathogens. Okay. So this AFM image very specifically shows that a DNA molecule. I don't know if it is visible on your computer screen very clearly. This AFM image we have done, this very clearly shows that the DNA molecule is anchored between two carbon nanotubes. So the second platform I quickly discuss. In this case, we have made an array, and instead of having two planar electrodes in front, we have this vertical stack. The beauty of this stack is that this can be fabricated in standard clean room wherever we have, and this can go into the manufacturing processes. In this case, what happens is two electrodes are separated by this blue, which is basically an insulator of nanometer thickness. And when the green one is target, so when the target is present, this is bridged across these electrodes. And we had a very simple metallization process to bridge the electrodes so that the current is flown. So in absence of this green, that is the target, we won't have the bridging of the electrodes. We won't have any signal. So this is the distinction. I won't go into the detail of this. I can only say that this particular platform is capable of detecting a single base mutation. A single base mutation is very important in the context of cancer, which basically create what the doctors say butterfly effect. That means a single base mutation can lead to a very serious consequence, including tumors and cancer. So I won't get into the detail. All right, so this is the overview of the biochip system that we have developed in collaboration with Procter & Gamble. It is protected by this patent and has been transferred to the Procter & Gamble Healthcare. All right, so coming to the last two slides of my talk. So far we have seen that we could do the fundamental biophysical studies and also fabrication of the devices. Now, in Indian scenario, I find that there is no sophisticated clean room available in abundance. Some in research laboratories, but not in the industrial settings. So here, what I want to convey the message is that we're trying to simplify the fabrication. This is fabricated in-house using our very modest and simple clean room facility at our Central Glass and Ceramic Research Institute. This is a simple array of interdigitated electrodes, and we are mounting it on a USB type of chip so that it can be interfaced with the smartphone kind of thing. And we have developed very small, as you can see the dimension. It is barely like 3 to 4 inches in dimension. So we have made this particular electronic signal processing circuitry that you can carry in your pocket. You have the smartphone by default in your pocket, and you can interface our sensor. This is in the end, the third picture, right hand side. You can see, you can take out the sensors, you can plug it into your dongle, and you can interface with the smartphone, and then you apply your sample, and you should be able to see the concentration of the molecule on your concentration of the target DNA, if at all present, on your smartphone. And with this, I had to conclude my talk because of the limitation of time. Thank you very much for your patience. And yeah, so this is now open for discussion if there is any.
Thank you, Dr. Somenath. I'm very sorry to interrupt you because we have
no, it's okay.
Timeline. So it is really very enlightening lecture, very informative. So if a couple of questions can be taken up. Any questions? So if there are no, maybe it is a little higher level DNA purely. So
no, I think if anybody had any question, then I think I can be contacted by phone, email.
Yeah,
we can discuss about it later on.
Maybe in the last minute, I have one question. Dr. Annapurna, can you allow?
Please.
Yeah. Actually, Dr. Somenath,
Yes.
I mean, the current strategies by which the COVID virus the detection and all, which is being undertaken,
Yeah.
how, I mean, how better way, I mean, how much better way your system, I mean, is there any possibility to tackle the current strategies, like in your case? Is it, I mean, the biomarker detection which you have done, which you have undertaken by your system? So
Mm-hmm.
can it be applied to the current strategies, or is there an improvisation in the methods, or will
Okay.
it be easier? Will
Yeah.
it be easier?
Okay.
Because there is requirement, no? There is a lot of
Yes. Yes.
requirement. Um,
Yeah, I think this is a very, very important question. And when our respected Director General is in our institute and I presented the last two slides, I mean, in association with other slides to him. So he immediately told the same thing, that can it be adapted to our COVID situation. And immediately we wrote a proposal, and we are in collaboration with CCMB to further this. So now from technical point of view, that's the management side. From technical point of view, I can say that this particular system has got two distinct advantages over the RT-PCR. The first one is that there is the first one, by name, it is PCR free. You do not need to have amplification. The kind of sensitivity that our sensor can achieve, you don't need to have all those thermal cycling, which takes quite a long time. And another very important part is that the sample leveling, since it is electrical method, you don't have to level the target. So leveling a target is definitely not point of care. Leveling means you need an expert to do the labeling of a DNA molecule. So that part also can be mitigated. So these two very important steps in RT-PCR can be totally obviated. So I can draw the analogy with the dry swab that CCMB has developed. There also they don't need to have those viral transport medium and RNA isolation. So two important steps they obviated, but still they are doing RT-PCR, that is direct RT-PCR. In our case, we are detecting by electrical methods, so we do not need amplification, no thermal cycling, and no sample labeling.
Yeah, I think that much of sensitivity. I mean, is it up to the mark? I mean, I have this question as well. The sensitivity, how it is? I mean, is it very good, or
Ma'am, we can detect down to femtomolar. The sensitivity is extremely high, and as I have demonstrated that some of these platforms, of course, there is some balance, the cost versus the accuracy. If you can afford to have higher cost, then it can detect up to single molecule for sure, because the platform has been designed in such a way the steric hindrance prohibit any two molecule to be captured between the electrodes and breach the gap. So that is there in the fundamental design consideration.
Okay, thank you, Dr. Somenath.